This is an excerpt of a conversation between J. Craig Venter and Emil Jacobs, brought to you by the Chemical Heritage Foundation's annual T.T. Chow Symposium.
So it suggests that you're not going to be using some of the technologies that would be normally used in E. coli and other species to introduce new genes. And therefore, you might not have the worries that people have about doing genetic manipulations of food and thereby introducing perturbations into the environment that we really don't want, since these algae could get into the environment and grow there. Is that a misrepresentation or not?
A little bit, but it's a very key issue. So I think we sort of had the view from the beginning that we would have to have either a very seriously genetically modified version of a natural organism, or primarily in my view, and when we started the alliance, we hadn't achieved the first synthetic life form. So, but the agreement does include the possibility of trying to make a synthetic cell. We don't, it's not part of the current research track. But either way, we don't want algae that produce 5 to 10 times as much lipid as normal algae to be released into the environment. It could have a real adverse effect on the environment if they could survive. There's been tens of millions of experiments done on laboratory E. coli, and there's putting genes of every species known in it, and there's not been any problems. And why is that? There's a chemical dependency for E. coli where growing it in the lab, if that chemical is not present, it can't survive. And so a key part of genome design, whether it's from modifying organisms or designing new ones, is to build in these safety mechanisms. So they either have to have a chemical dependency if they're going to grow in an open pond, or we're designing all kinds of new mechanisms, self-destruction mechanisms. And in fact, when the Obama Bioethics Commission was charged by the president last year following our announcement of the first synthetic life form, a big part of their report is urging the scientific community to develop more of these safety mechanisms that could go into a variety of cells. So we have to have it so if something's ever released accidentally or deliberately into the environment, it can't survive, period. And so, you know, it's building in the safety mechanisms from the very beginning as part of the design. In fact, we're trying to enforce it as part of the ethos of this entire field. We watermark the DNA. We can actually be clever with it. We developed a new code where we can write the complete English language in the genetic code. So any organism we can make sure that it's labeled. It'll say either ExxonMobil SGI or SGI ExxonMobil.
That's the one you like.
We'll probably debate that for a while.
I'll give you that one.
And we can have these mechanisms built in. So you can track to see if it got out versus somebody thinking they made some discovery of some natural algae that really produces these lipid levels. But safety is really paramount to this. Otherwise, this new industry can't possibly survive.